Hello dear student, today in this lecture we will discuss about the monoclonal antibody, importance of monoclonal antibody and the production of monoclonal antibody and monoclonal antibody therapy in COVID-19. So let's start our lecture that how monoclonal antibody can help in the COVID-19. Before starting our lecture on monoclonal antibody, first of all, we must have to know about what is antigen. Antigen is a molecule that can initiate the production of antibody and causes an immune response. Any molecule will enter the body and they initiate the production of antibody. This molecule is called antigen. Now this antigen are mostly protein present on the surface of pathogen such as bacteria, fungi and viruses and other foreign bodies. When what are antibodies? Antibody are produced against this antigen, which antibody are produced against this antigen when they enter the body, and actually they are polyclonal. The antigen are different, so different type of antibody are produced when different antigen are coming to the body, and different type of antibody are produced. These different antigen, these antibodies are called polyclonal antibodies. And you know that I want to give you example. Suppose this is a bacteria and this bacteria have different antigen. This is antigen number 2, antigen 3 and antigen 4. Now against this each antigen different type of antibody are produced by the body. So we say that our immune system response is polyclonal. What does it mean? It means that when a bacteria come to the body, these bacteria having different antigen, and against each antigen different antibody are produced. So our immune response is polyclonal in nature because microorganisms are mosaic. One microorganism have different type of antigen. So for example, if this bacteria come against this bacteria is having different structure, so against this bacteria different antibody are produced. Now polyclonal antibody, they are not specific. They can bind with the any type of antigen. Now what is monoclonal antibody? Monoclonal antibody, the type of antibody which is produced by single cell or clone of cells. Actually these antibody are genetically engineered antibody, and they can give response, they can recognize a specific antigen, and actually it provide passive immunity. What does it mean? It means that when we produce the monoclonal antibody, this monoclonal antibody will not give response to all antigen, but it will give response to a specific antigen, and it actually produce a passive immunity. Now how does monoclonal antibody therapy work? Actually monoclonal antibody therapy, it is also called monoclonal antibody infusion treatment. It best work in the treating of COVID-19 coronavirus. Now the goal of this therapy is to prevent hospitalization, reduce viral load and lessen symptom severity. Now the question arises that what does it mean? Actually we made monoclonal antibody against the spikes of the coronavirus. Coronavirus have spikes. What we do, we produce against these antigen, against these spikes antibody, and this antibody only attach with the spikes of the coronavirus. Monoclonal antibody, actually they are mass produced in a laboratory and are designed to recognize a specific component of this virus. The spike protein and its outer shells, they target it. Monoclonal antibody therapy is given through intravenous infusion, and you know that you should do monoclonal antibody therapy for the coronavirus. What does it mean? It means that when the virus attack, when the virus have enter your body, you can use monoclonal antibody, and this monoclonal antibody specifically bind to the virus protein, and in this way it will kill the virus. Now the question arises that how you can create the monoclonal antibody. The first step, that create antigen of interest against you want to make monoclonal antibody. So you should first create your antigen. Suppose if you want to make monoclonal antibody coronavirus, you should make the antigen of that virus. If you want to make a monoclonal antibody against tumor cell, you can make the antigen of the tumor cell, any type of cell. What you will do, use the mouse to inject, use the mouse, and you inject this antigen to this mouse. This is mouse. You can inject your antigen to the mouse. You can inject the antigen to the mouse. What will happen? This mouse will immunize. It means that immunization of the mouse produce B cell in the spleen. Mouse B cells, mouse spleen have spleen in which B cells are produced. Now when these B cells are collected from the immunized mouse and cultured for a short period, but B cell cannot survive for more than a week because it is mortal cell. Now this is a big problem. How you can produce large amount of the antibodies? So the solution is that you can use hybridoma technology. In hybridoma technology, what we can do, we use two type of cells. What this type of cell? The one cell we taken from the mouse spleen, that is the first cell. The first one is B cell from the mouse spleen, that is mortal, and fuse them with the cancer cells, I mean the immortal cell. You fuse them with the immortal cell. You fuse these two type of cell. What will they do? They will form hybridoma. Hybridoma mean that it will contain the B cell of the mouse as well as the cancer cells. Now this hybridoma cells, they have the ability to produce antibody as well as it is immortal, not for specific time. Now this hybridoma contain different type of cells. Myeloma cell, they contain myeloma cell, they may not be fused, and they can also contain hybridoma cell, that is our choice. To separate the undesirable cell from this media, from this culture, we actually grow this on a special type of media called HAT. HAT contain hypoxanthine, aminopterin, and thymidine. What will these HAT media do? These HAT media will selectively separate the hybridoma cells. And what we will do, this hybridoma, and from this hybridoma cell we will collect our desired antibody, and this antibody we will use for different purposes. Now what is the importance of monoclonal antibody? A monoclonal antibody can be used to detect pregnancy only 14 days of conception. Monoclonal antibody allow rapid diagnosis of hepatitis, influenza, herpes, streptococcal, and chlamydial infection. Monoclonal antibody can also be used to purify a substance with a technique called immunoprecipitation and affinity. Monoclonal antibody can also be used to treat cancer.